So our next speaker, Eleni Mimitou, received her doctoral degree in genome integrity from Columbia University, where she studied DNA damage repair pathways. During her postdoctoral training at MSKCC, she developed a sequencing-based assay to find high-resolution maps of DNA repair intermediates. Last year, she joined the Technology Innovation Group at the New York Genome Center, and her current focus being on single-cell multi-omic applications for cell characterization.
Thank you very much, the organizing committee, for giving me the opportunity to represent our group today from the New York Genome Center. Special thanks to Adib for delivering almost. I was planning to give an introduction on CITE-seq and cell hashing, short, but still. So now I think I'm going to go a little faster since beautiful talk is still fresh in your mind, and mainly focus on the CITE-seq extensions that we have been recently working on. I'm sure you're convinced by now how both methods to interrogate complex cell populations have strengths and biases, and by combining them, we get a much better chance of interrogating these complex populations. And to do that, we use antibodies to detect cell surface markers, and we turn these antibodies into something that the sequencer can read, but just making it look like an RNA. With an oligoconjugated that contains a poly-A tail that mimics the RNA, an antibody specific that works as an identifier for the antibody, a sequence that acts as an identifier of the antibody, and a PCR handle that allows specific identification of the scalability that we frequently have to face with CITE or flow cytometry is not an issue anymore. The barcode space far outnumbers the antibody regions available. This was the idea how it was born about 2 years ago. CITE-seq stands for Cellular Indexing of Transcriptomes and Epitopes by Sequencing. Again, I described how it's performed. The main idea is that depicted here in droplet-based single-cell RNA-seq methods, in the beads where the oligo-DT is bound, upon lysis, everything that is poly-A tailed in the cell is captured. So along with the mRNA, we capture the oligos that are antibodies, and they're all indexed to the same cell barcode. Because of their vastly different sizes, we can separate them out and make two different libraries that can be and get information for both the transcriptome and the surface markers. In this study led by Marlon, we used immune cells as a way to benchmark CITE-seq. They are very well characterized by both single-cell RNA-seq and chemistry, and there were a lot of markers that are highly specific, and antibodies, a variety of antibodies available. And again, with the data that was already shown, I'm sure you're convinced that the CITE-seq allows fast detection that is not subject to dropout events. It can allow us something that maybe it didn't touch on so much, that allows post-transcriptional regulation at single cell level. I'm going to draw your attention to two examples here where I hope CD11C stains the RNA transcript is present on the NK cells, but not the protein, surface protein. Similarly, CD16 is present on these CD16 monocytes, mRNA, but not the protein. And then with the data that we showed and that Marlon showed in that study, we can reveal phenotypes that cannot be discovered by just using. Moreover, an extension of CITE-seq that was very enthusiastically received is cell hashing, which is essentially multiplex CITE-seq antibodies with cell surface markers in a manner similar to CD40 multiplexing. So this is a way to, we can stain different samples with barcoded antibodies. We also use the cocktail of B2M beta microglobulin and ATPase, the cell surface ATPase, 8298. The samples being stained and washed are pulled into the same run, and we can demultiplex the sample later using the barcode and trace each cell back to the sample it originated. So this can be used, as Adib said, as a way to multiplex runs. Sometimes we have only a few cells that, from a tumor biopsy, let's say, that it would be a waste of money to run them only in one 10x lane. Now we can pull many of those samples together and get a lot of out. Also, it can be a way to overload a run when we have a lot of cells available. And in this case, confident double identification allows us to discard them and focus on and again, as I did touched on, one application that I find really useful is that we can use hash tag information as a way to filter to retain cells that were discarded as low UMI cells, but they're actually they have important information. In this example, there's about 3,500 cells that were considered like low UMI cells that could have been empty droplets. But when we look at HTO counts, so if they're positive for hashtag information, you see that they cluster into canonical blood groups, suggesting that were real cells. Whereas if we look at droplets that were negative for HTO information, they don't group into any clusters, suggesting that they were real, probably ambient RNA in an empty droplets. So to summarize this part, oligo-conjugated antibodies allow integration of protein RNA-seq. Integrated protein and RNA measurements enhance cellular phenotyping, and cell hashing allows sample multiplexing and so my main focus when I joined the lab was how we can further extend CITE-seq technology. I'm going to move on to the new extensions that we have recently developed. In most single-cell RNA-seq workflows, a key step is the reverse transcription of the mRNA primed by a poly-DT RT primer on the 3' on the transcript, which extends to the 5' on the transcript, and then performs a template switch with the template. You can envision either of these elements can be barcoded and bound on the bead. In what most traditionally is done is that the barcode, the poly-DT primer is barcoded and bound on the beads, and in that case we get three prime tagged libraries. 10x Genomics now offers the alternative, but the oligo is barcoded, and in this way we can make 5' tag libraries. Now the RT primer is soluble. So that gives us the opportunity of doing selective enrichment of 5' values in the context of high-throughput single-cell RNA experiments. And that was first exemplified with the reconstruction of rearranged VDJ regions of immune receptors sites. Here, by using the full-length cDNA as it comes out from regular protocol, and using specific primers against the constant regions, you can amplify transcripts that contain information about the TCR alpha beta or the heavy chains, and get information in a single cell level for the paired receptors. So we wanted to combine that with CITE-seq, and overall we thought that the ability of this, the opportunity of using this solution is much more customizable because soluble RT primer can open up many other opportunities. So to make it compatible with CITE-seq, we just modified the oligo that is bound on the antibody And instead of a poly A tail, it has the sequence complementary to the switch oligo that is hanging off the beads. So during RT, it can anneal to this part of the oligo, extend and copy the cell barcode and the this way we can make again libraries of the oligos in a similar way to the 3' CITE-seq. And also you can imagine a similar structure of the hashing antibodies where the oligo bound on the hashing antibody only has a amplification of this. To see how 5' CITE-seq performs, we used PBMCs from a healthy donor via our collaborator at NYU. And we stained with a panel of 49 5' compatible CITE-seq antibodies and 4 5' cell hashing antibodies. First, I'm showing you clustering based on the hashtag information. So you can see we retrieve the 4 clusters, each one corresponding to its hashtag used, and 6 smaller clusters corresponding to the doublets, which we can remove and focus our analysis on the sequence. Here on the left, I'm showing you transcriptomic clustering of healthy PBMs, which fall into the canonical groups. And on the right, I'm showing you a comparison between transcript and levels on a handful of markers. No, there's no time or bandwidth markers. And as noticed, as we've seen before, for 3' CITE-seq, there's a very good correlation. The detection using protein is much more robust. It's not subject to the dropout problem. So if I take it from the beginning, CD4 is present in the CD4 cluster. There's a good separation with the CD8 decoration. HLA with DR, which is to be found in monocytes and B cells. Indeed, CD11C mainly in monocytes, CD27 T cells and memory B cells, and CD19 mainly in. We do face sometimes some background staining, and sometimes it's experiment dependent, but there are ways to control for that. We have some internal controls added into our experiments by spiking in different species cells and having some IgG control. But as I mentioned before, the point, our goal was to combine CITE-seq with PCR reconstruction. So here in the same sample, I'm showing you side by side those 2 CITE-seq markers with the PCR alpha chain transcript compared to the top 2 chronotypes out of about 2,800 total. And they are, of course, they're present where they're supposed to be. And we took it a step further, and by using custom-made primers against gamma and delta chains, we can also reconstruct this delta receptor. And here shown again the top 2 present in 18 and 15 out of about 207. Expected that they would be much less than the alpha beta. Now, to showcase the strength of bringing all these modalities together, I will just share a vignette of a recent CITE-seq experiment that we performed, again with our collaborator Sergei Kovalev, NYU, and Anthony Cheng at Jackson Laboratory. So here we have PBMCs from cutaneous T-cell lymphoma patient, alongside with the control PBMCs I showed you before. This is transcriptomic-based clustering after cell alignment using the recently published PGL pipeline. And first colored, I'm showing you the cell identity, what's coming from the healthy. Here is presence of a productive chronotype as detected by the TCR alpha beta Here is CITE-seq for CD3, just an example. It shows good correlation with the presence of the active chronotype. And these are the top 2 clones detected in the malignant population and the healthy population. And you can see the clonality of the top chronotype in the malignant cells compared to the healthy cells. Now, in silico gating based on CD3 and CD4 levels combined with determination of the malignant chronotype allowed us to focus on these 3 different groups of cells. The malignant CTCL cells, the normal within the malignant sample, and the control CD4. And as you can see, the transcriptional profiles are vastly different. This is just a small example of how we can, by merging these 3 features, chronotype, protein, surface protein with CITE-seq, and transcriptional readout, we can achieve a much deeper characterization of heterogeneous cell population. Moving on, another avenue that we wanted to explore, and that's made again feasible by the 5' protocol because of the custom, the RT primer being soluble, is direct guide capture, guide RNA capture. So again, as Brian very well presented a few hours ago, it becomes obvious how when we do CRISPR pool screens, how important it is to get a readout in a single cell level. And lately, a lot of groups reported using single-cell RNA-seq as a readout to gain better information about how the perturbations are mediating their effect. The 2 major approaches until now were using Perturb-seq and Prop-seq, which both rely on placing a guide under a longer Pol2 transcript and either sequencing an associated barcode or the guide itself as part of this transcript. The issue with Perturb-seq that was recently covered is that there's very often, and as often as half of the cases, there's a swapping between the barcode and the associated guide RNA, and that can result in erroneous conclusions. And with Prop-seq, one issue was the low capture rates because it's at the end you capture a transcript, a Pol2 transcript, so it's subject to the dropout problem. This was partially resolved. It was actually very well resolved by just doing an enrichment of the transcript that were with specific primers. But again, the problem remains the same. You still have to clone your guides on specific vectors, and it cannot. When you already have a library, you have to go back and remake it so that it's compatible with this style of capture. We wanted to achieve a direct guide capture, and for that reason, we leverage the invariable scaffold sequence of the guide RNA as the priming site for a guide RT primer that we can spike in during the RT reaction. This extends and then same alongside with the mRNAs in that cell can do template switch and copy the UMI and cell barcode information. Once now the guide information is in the same string, the cell barcode and the UMI, we can PCR amplify and make the guide-derived oligo library. To see how this performs, we did a proof of principle experiment with a small number, 9 non-targeting guide RNAs that were transfected in 3T3 mouse cells. These cells were stained with 7 barcode hashing antibodies and then went through the 10x 5' protocol. The only difference being that during RT we spiked in the guide RT primer. After spike size selection, which is a normal step during this protocol, the bead bound population, which is the longer ctDNA population, is used to make the ctDNA library. whereas the supernatant contains the shorter oligos coming from the hashtags and the guides. And they can be used for further amplification to make the libraries. Here I'm showing you clustering based on the HTO information. And you can see that we retrieve those 7 main groups, plus some smaller groups of the doublets that can be removed. And once we remove them, we can ask, now that we know that we have single cells, we can ask what are the capture rates for our guides. And we see that the capture is pretty high, only 6.5% of the cells were negative. And from the positive ones, about 85% had 1 unique guide per cell, and about 8.8% had 2 guides per cell, which most of them are just singlet cells that have expressed 2 guides. And because we remove the doublets, we can now be more confident about the information coming from these cells. Moreover, when we, again, focusing on singlet cells, when we look at, we see a good anti-correlation between the GDO counts, suggesting that the single cell capture happened while the cells were in a single state. And when we look at representation, all of the guides were there, albeit with a different distribution. This is demultiplexing based on GDO counts, and then assigning each cell to a guide. But in fact, that mouse sample was only part of the sample that was taken into this 10x round. It was only 65%, the 25% were PBMCs, and 10% were some lymphoma cell lines. These were stained with 5' compatible mouse and human CITE-seq antibodies, CD29 to be more specific, and then run in 1 lane of 10x. And after price selection, we use the bit fraction to make cDNA libraries, TCR alpha beta and gamma delta libraries, and the soup to make ADT library, HTO, and GDO library. So here I'm showing you transcriptome-based clustering, and the big population is the mouse cells, that was 65% of the sample. The smaller and again overlaid colored highlighted is the human hashing counts. Purple for the PBMCs, orange for the myelin lymphoma cell line, and green for the Sézary cell line. Because of the different gene expression profiles in the cells, they separate very well, also based on transcriptome, but it was very reassuring to see that the hashing information agrees completely with that separation. And of course that the mouse cells were largely negative. Now, we wanted to overlay all the information we had for each single cell in this experiment. This is the CITE-seq staining, specific for the species, mouse CD29 stained mouse cells, human CD29 stained human cells. Guides were only present in the mouse cells, as expected, and finally, TCR alpha beta and gamma delta were present in the PBMCs and the 2 lymphoma cell lines. And again, you see the clonality, so that these are the top 2 clonotypes for the 2 lymphoma cell lines, and they're much more clonal in these cell lines than in PBMCs. So to sum up, we are literally very excited to announce CITE-seq, which stands for Expanded CRISPR-Compatible CITE-seq. In this single assay, we can get from single cells information about gene expression, TCR alpha beta gamma delta, BCR, even though I haven't shown it yet. We can get antibody information in the form of CITE-seq and cell hashing, and we can get guide information by direct guide capture. And this is actually a very exciting avenue we're currently exploring, because it paves the path to detecting any non-polyadenylated transcript using custom primers. And to conclude this part, CITE-seq and cell hashing are now compatible with both 3' and 5' 10x technologies. CITE-seq and TCR reconstruction can interrogate complex cell populations in a deep and comprehensive manner. Direct capture is possible with a 5' and greatly simplifies using single-cell RNA-seq and CITE-seq, particularly as a readout for pooled screens. And CITE-seq allows single-cell capture of at least 5 modalities of single assay. It depends how you count. I tend to say 7 modalities because it's 7 libraries that I have to prepare and submit for sequencing. So with that, I would like to thank the Technology Innovation Lab, an amazing group of people. I'm not exaggerating. I was convinced CITE-seq works well, but after your talk a bit, I'm in love with it. I would like also to thank our collaborators, especially Sergey Korolov at NYU and Anthony, who did the analysis on the combined CTCL and healthy donor PBMCs. Rahul and his people and neighbors on Genome Center, and Neville. So these are in-house single-cell RNA-seq and CRISPR experts. And funding, BioLegend for CITE-seq and cell hashing antibodies. All the information, almost all the information I presented can be found in our website with Peter keeps it continuously updated. And please follow us at Twitter. Thank you for your time, and I'll be happy to take questions.
Talk about or ask where you need to guide for the gene target or knockout.
So that's the next.
Very
This
nice.
was long time.
Beautiful. I mean, we have tons of libraries sitting in our freezer that now we make use of.
That's the thing about it. We get like any existing
Yeah,
library.
exactly. Yeah.
No need to construct them. So the proof of principle I've done is. Oh, sorry. Yeah. The proof of principle I presented was with non-targeting guides. I'm working now with a library that has targeting guides, and a collaboration we are currently working with in Rahul's lab also had targeting guides, and but we never explored this. It's part of our plan to look at the mutation itself and the perturbation.
If you give me one where you could do CITE-seq on the, sorry, I used to say, you know, where you could do the antibodies, those markers, and do everything well.
So that's the experiment I'm doing almost right now. But I'll be happy to do it in a much bigger. So the guides I have now are about 20. So if you go into hundreds, and I will be very interested to try and